you, Bob. Well, today it's my pleasure to introduce Dr. Josef Uyeda. He's an assistant professor at the Department of Biological Sciences in Virginia Tech. Josef graduated with a PhD in ecology and evolutionary biology from Oregon State University. He was advised by Steve Arnold, and his dissertation focused on building across microevolutionary and macroevolutionary scales, but also spatial and time constraints. After that, he moved on to be a postdoc at University of Idaho in university with Luke Harmon. One of the reasons why I invited Joseph to give this talk is because I met Joseph as a postdoc in Idaho, and having him at arm's reach, just one desk, approach models in macroevolutionary biology. But I must say that most of the people that have met Joseph or have talked to him in science or life have the same experience. His thinking is very, very changing and has been full force in my field. But I'm not only saying this from personal experience. I think NSF agrees with this claim. He was just funded last year, his NSF career, with a project that is integrating causal evolutionary processes into phylogenetic comparative biology. And with that, thank you, Joseph. I'll pass it to you. We're very excited to have you here today.
Thank you very much, Rosanna. That was a really kind introduction. I'll try to live up to it. And thank you all for joining today and taking some time from hitting refresh on your browsers to listen to me. So let's see, I want to start my presentation here, and then I will go ahead and share my screen. All right. Okay. And you see the slides there? Okay, great. So what I want to talk about today is macroevolutionary causes. And so how I want to start this talk is by thinking about how we study causes in evolutionary biology. So we might be interested in what causes this phenotypic change, say a body size change in Galapagos finches. If you're familiar with Peter and Rosemary Grant's work, you probably have some understanding of what sorts of things can cause this type of change by evolutionary processes. Things like droughts causing seed availability to change, causing beaks to get bigger or smaller depending on the relative frequency, things like that. But we're also interested in what causes this, what causes the diversity we see across the tree of life. Why do we see all these different forms? And this is a much greater scale. And we can ask the question of what causes this, and I think it's a tall order, but it's also a fundamental and an essential question. Are the causes of these things the same causes that we observe on the microevolutionary scale? And whatever your answer to that question is, I want you to think about how strong is the evidence for that. How do we study that? How do we understand? Where is the evidence for the causes of evolution at this scale? Of course, we can't do experiments, but it's vital that we understand this because this is the relevant scale for understanding adaptive radiation, stasis, extinction. And if we don't understand the causes of variation that we see across the tree of life, what hope do we have to have meaningful interventions in the face of global change? So what tools do we have to address these questions? Well, Harvey and Pagel in 1991, building on work by Joe Felsenstein years earlier, wrote this, that phylogenies help us to identify independent evolutionary events, and it is independent events that statistical tests rely on. And so the idea here is that, okay, experiments may be impossible, but we can identify independent evolutionary events and do statistical tests using the phylogeny. And so I think what was taken from this is that we have a phylogenetic comparative methods toolkit that we give to biologists who are interested in asking these sorts of questions. And in the decades since Harvey and Pagel wrote those words, the phylogenetic comparative method toolkit has expanded dramatically with all sorts of models, all sorts of statistics. And I think it can be a bit overwhelming for students and empiricists who are interested in asking questions about what are the causes of evolution in their particular group. And what I think the message from for proper use of this toolkit is that you can use this toolkit, but you have to be a little bit careful. The first thing you do is you throw out the word causes. You use the word drivers instead. If you look for causes of diversification, you won't find anything. If you look for drivers of diversification, that's in the title of lots and lots of papers. And if you just remember these things, you can have a successful inference. You should correct for phylogeny. You should get all the data you can, get more and better data. And make sure that your assumptions of your model are satisfied. And above all, remember that correlation does not equal causation. And as long as you keep all these caveats in mind, you can move forward. And I don't disagree with any of these things. But my thesis for this talk is that what we really need to make good inferences is mostly absent from this toolkit that we're providing. That we need to consider causes. We need to consider cause and effect. The key, and the key to all this, I think, as I say, it's kind of intimidating, all these different models, all these different statistical techniques. I think the key to good macroevolutionary inference is not putting more of these, teaching more of these things and putting them all into students' brains, but putting more biologists' brains into our analyses. I like to use this quote from Judea Pearl. I recommend strongly this book. I've been kind of suggesting everybody read this because I think it will change, potentially change your thinking on how we do a lot of these studies in macroevolution or in evolutionary biology generally. And I love this quote. We live in an era when big data is presumed to be the solution to all our problems. But the one thing I hope to convince you is that data are profoundly dumb. If I could sum up the message of this book in one pithy phrase, it is that you are smarter than your data. Data do not understand causes and effects. Humans do. So what I want to start with is how causal inference can affect what we do in phylogenetics. And I'm going to give a simple toy example for that. And then I'm going to go through a few case studies where I outline how causal thinking has influenced my own work in phylogenetic comparative methods and made me do things that I might not have done otherwise. Okay, so let's start off with causal inference in phylogenetics. So in the early days of phylogenetic comparative methods, there was some pushback against this idea that if you have comparative data, you should always have to use the phylogeny. So this paper by Mark Westoby and colleagues argued that we shouldn't always do this phylogenetic correction. And they have this figure where they kind of explain their argument that if you have some percentage of trait variation correlated with phylogeny and some percentage of trait variation correlated with ecology, we're interested in what's correlated with ecology. But if we correct for phylogeny, we're going to attribute some of that stuff that's correlated with ecology to phylogeny, and we only have a little bit left over. And that's because ecology also follows the phylogeny. Now, this, and so they were arguing that we shouldn't always correct for phylogeny in this case. And the response to that, which I think is kind of what stuck, it's what I learned, it's what I think a lot of people learned, is this response by Harvey, Reed, and me. They repeat the claims, and then they just give this one-word response to it, that it is wrong, and kind of left it at that. And I think, I agree that the argument in Westoby had serious flaws in it, but the overall spirit of that argument had a lot of merit. And I think that by dismissing it out of hand, we've potentially made some mistakes in how we think of this. And in those early days, it was well identified that that phylogeny is simply a case of Simpson's paradox. And so do you take the regression within these groups, or do you take the regression between these groups? And I'm going to give you an example of Simpson's paradox and let you guys play along. So imagine this is observational data. Okay, so this is not an experiment, but we have two tables here where a drug was taken by some people. For the male table, if there was no drug, they recovered 70% of the time. If they took the drug, it was 60% of the time. The female recovery rate, the drug was 20% recovery rate, no drug, 30%. Would you take the drug in this case? Well, it doesn't seem like it does a very good job for either males or females, so most of you probably would not take the drug. But if I just combine these data, I get this result, that with the drug, you get a 50% recovery rate, and no drug, you get a 40% recovery rate. Now it seems like it's a good thing to take the drug. So would you take the drug or not? So if I was in a room with you, I'd have you all raise your hands, but hopefully you're all logging in your answers. And so this is Simpson's paradox, and what you do here has been written extensively in the statistical literature. But the most compelling explanation for what the answer is, whether you should take the drug or not, really comes not from statistics, it comes from your assumptions about cause and effect, okay? And to demonstrate that, you can take, this is a figure from Pearl's book again, is that most of us are probably thinking of gender as a confounding influence on this analysis, right? If instead of male and female, we had high blood pressure and low blood pressure, things might be a little different for one specific reason. You probably don't think that taking the drug will move you from one table to the other. It won't change your gender, right? That's generally not something that you want a drug to treat a disease to do. But if it was instead high blood pressure and low blood pressure, because it's an observational study, taking the treatment may move you to a high blood pressure or a low blood pressure category, which then may be the mechanism by which treatment and recovery is enacted, right? So in the case of the former, you would want to use the gender-specific tables. In the case of the latter, you would want to use the combined table. And it's entirely dependent on the causal effect. We can do the same thing for phylogenetic studies. So this is an example that was kind of the basis of some of these early arguments. This idea that we wanted to see a relationship between body size and population abundance. So body size has a phylogenetic effect, suppose. It evolves down the phylogeny, but we actually observe body size, and we're interested in its effect on population abundance. Maybe if you get big, if the species increases in body size, because each individual takes more resources, it's reasonable that population abundance may go down. But there's another effect on an unobserved variable, and we're going to call this migratory behavior. So as you get big, maybe you get the itch to migrate more, cover more ground, and so that you can actually use more resources, which then allows you to increase in population abundance, okay? Compare that to this causal model where migratory behavior has a phylogenetic effect, is unobserved, and also affects not only population abundance in the same way, but also body size. So here are two alternative causal hypotheses. Under this, and so what you should do with your analysis here depends on which of these models is true, which of these assumptions is made. And the key to unlocking this is to think about experimentally manipulating body size, okay? Artificial selection or something. So we artificially select on body size to increase under this model. What we're gonna do is increase or decrease population abundance, but we're also going to cause an increase in the probability that a lineage transitions to becoming migratory, which then may increase population abundance. So manipulating this system might have two contrasting effects on population abundance. In this case, even if you have phylogenetic data, you should take the non-phylogenetic regression. part of the reason you can see that is because you observe body size. It blocks the effect of phylogeny because you actually observed it. You don't need to know that it has a phylogenetic effect after you've already seen it. Contrast that with this model where you artificially select for body size. You're going to change population abundance, but you're not going to affect migratory behavior. Migratory behavior has a phylogenetic residual effect. So then in this case, you would want to take the phylogenetic regression, which would be within clade, because you're not going to move between those two different groups. Now, reality is probably potentially some combination of this, but it just illustrates this effect that we shouldn't always just assume that we should automatically use phylogeny, which is what, at the heart of it, Westoby's original argument was in those early days. And so should I include confounding variables like phylogeny in my analyses or not? And so I'm going to talk about an example that recently, a project where this came up for us. And so this is a project that was led by Vinny Ferrallo, who was a postdoc. He's now assistant professor at the University of Scranton, and Don Miles and Martha Munoz. And they were interested in salamander evolution, okay? And so in particular in response to climate. And so Vinny was really interested in how we measure these climate preferences. The typical easy thing to do is to just take museum records for a species, get the temperature and precipitation for a grid cell on Earth, and kind of use that as data on their climate niche, right? There's reasons to think that that might not be very good for salamanders, which choose very specific microclimates in a forest that might be very different from the prevailing macroclimate. And so what he did is he went around collecting microclimate data on a bunch of different species of salamanders, on temperature and relative humidity at the site of capture, and he analyzed both of these data sets. And sure enough, there was a very strong phylogenetic signal in the microclimate data, but no significant phylogenetic signal in the macroclimate data, suggesting that the microclimate data was capturing some evolutionary signal of temperature and humidity preference. But we weren't just done there. We wanted to see if the macroclimate changes, does the microclimate eventually follow along? Is there a relationship between these two? And so we did a phylogenetic regression analysis using the package Slouch. I won't go into the details of how this analysis works, but basically we can take the macro scale as a predictor and the micro scale as a response, and we can see the observed evolutionary regression. But this analysis also gives us an estimated optimal regression, which is interpreted as if lineages were allowed to evolve for an infinite amount of time, what would the eventual relationship between these two things be? There might be an evolutionary lag between the macro scale temperature and the microclimate preferences catching up. Okay? The dotted line is the 1-to-1, a slope of 1, okay, which would be where the two were equal. And you can see that our analysis suggests that if salamanders are allowed to fully adapt in the same place with a constant macroclimate, they will eventually reach that 1-to-1 slope, but there's a lag, probably because of evolutionary lag in adaptation to this new conditions. We can actually estimate that lag, and we found strong evidence that it's older, it's at a minimum about 17 million years, which is a long time. And we thought this was pretty cool because what this suggests to us is that instead of evolving when the climate changes, salamanders are just choosing different microclimates so that they don't have to evolve. They're choosing ancestral microclimates, and it takes 17 million years for them to finally kind of catch up to whatever the change in the macroclimate is. So we thought this was cool. We submitted it, and we got a review back. We got reviewer 3. And reviewer 3 says, you know, I looked at the sampling of your sites, and it wasn't random. Your microclimate signal could be an artifact. You should include date and time of day as covariates. So Vinny, it turns out, can't teleport. And so he can't actually sample sites randomly across the United States. Instead, he drove around as a grad student collecting salamanders and measuring temperature and relative humidity where he found them. And he couldn't do that all in the same month. In fact, it stretched out over several months. And of course, the temperature you measure is going to depend on what day and time you sample it. But I thought there's no way that this is a problem because what are the odds that he would drive around the United States in such a way to perfectly capture the phylogeny, right? That just didn't seem at all possible to me. But we humored the reviewers and we tried to do it anyway. And so we redid our analysis, but we included date and time of day as covariates. When we did that, we got no significant, it took away all of our signal. Worse than that, if I just took the days that he collected salamanders and went to weather.com, so absolutely nothing about the salamanders, went to weather.com, pulled down the climate and analyzed that on our phylogeny, there was strong phylogenetic signal in that data. So it didn't seem like it had anything to do with the salamanders. It was entirely to do with when Vinny went out and collected data. And so this disturbed me, and I was like, take me off this paper. This seems really bad. I don't want to do this. But we started thinking about it a little bit more carefully. And I sat down and I made a causal model of what I thought was happening. And I thought, there's one way that we can salvage this. We do this one test, okay? So I thought about it this way. Okay, so Vinny made life choices that determined when he went out to collect salamanders. And those determined the climate conditions. But in order to end up in the data set, somebody else had to show up that day. And specifically, in order to be actually measured, you had to have a salamander present. And the salamander has its own preferences, which is what we're actually interested in studying. So if you had an analysis where you took you had all the days he went out, not just the days he found salamanders, and did an analysis and there was no phylogenetic signal, but that phylogenetic signal emerged conditioning on salamander presence, then it would suggest that only through the salamander preference is how that phylogenetic signal emerges. But you'd have to be crazy to collect data for every rock that you flip that not only did have a salamander, but every rock you flipped that didn't have a salamander. But actually, that's exactly what Vinny did. He had data on every rock he had flipped over several years. And so we could actually do this analysis, this test of this hypothesis. And sure enough, if we take all of his site data and do a phylogenetic analysis, there is no phylogenetic signal in either relative humidity or temperature. But as we condition on salamanders being present that day, it starts to get a little bit higher phylogenetic signal. As we weight by the abundance of the day, we get very strong phylogenetic signal for temperature. And as we take direct site of capture data, we get marginally or significant phylogenetic signal. In other words, as we increasingly condition on the behavior of the salamanders, we see the emergence of the pattern. So it's not just date and time as confounding factors, but date and time are actually how the salamanders are expressing their preference. They come out on days of the year where the temperature fits their temperature preference profile. Otherwise, they're going to be underground and you're not going to find them. So our phylogenetic model not only predicted microclimate preferences, but it also predicted phenology. Phenology seemed to be a lot more important than the particular rock that they were under, right? They are able to control their preferences primarily through the timing of when they're active. And this stasis, or this inertia to adapt to macroclimate, appears to be driven by the Bogart effect, which is behavioral inertia. Their behavior is slowing down their evolutionary response to macroclimate because they can just change when they're active rather than evolving a response to a warmer or a colder climate. Okay, so we shouldn't always simply throw in our covariates, even if they're significant. Sometimes those things are the mechanism by which we should, that these things are occurring. Another example of a study where we were looking at this, how causal inference influenced our thinking, is a project that was led by my postdoc, now an assistant professor at Murray State University, Suma Nupani. And we were interested in plant growth forms in response to climate change. And there's some interesting traits that plants have that can be influenced by climate. For example, vessel diameter. Plant vessels in freezing environments, if they're too big, they can have freezing-induced embolisms, which can disrupt the flow of fluids in the plant. Think of basically a plant version of the bends, right? That breaks the conduit when you get these freezing temperatures. And so it might be important to have smaller vessels in a plant that has persistent stems in a cold environment. And so this is something that I had examined previously in collaboration with Amy Zan and colleagues, where we were interested in looking at the effect of temperature on vessel area. And we found a very strong effect that vessels got smaller as temperatures got lower. And also we found this interesting effect where evergreen plants, which are exposed to freezing temperatures with leaves, so they need to be transporting fluids when the temperature is below freezing, they actually have this sigmoid curve where they suddenly drop down right at about 0 degrees, where they have to drop down faster than a deciduous tree that loses its leaves and probably isn't transporting as many fluids when it's actually freezing temperatures. And so we find this causal model where low temperatures decrease vessel diameter, and there's this interaction between leaf phenology and temperature that also affects vessel diameter. Around the same time, a paper came out from Olson and colleagues looking at this same sort of relationship. And what they found is that vessel diameter is very strongly correlated to stem length, or the height of the tree, right? And that plant height seems to be the primary factor that's affected by the cold. So low temperatures cause shorter trees, and that in turn decreases the vessel diameter. And so you have these two possibilities kind of nested within this much broader, probably network of things that are interacting in plant growth forms. But we usually only study these one at a time. We study these kind of piecewise. And so you have these two stories here. Which of these is true, right? How do we reconcile these things? And so what we use is a phylogenetic path analysis. And so phylogenetic or path analyses are a form of causal inference. And it's true that you can't prove causation from correlation, but certain patterns of correlation can reject many causal models. And so what you're left with after a phylogenetic path analysis is a set of plausible causal models, okay? And so what we did was we tested 607 different possible causal path analyses and ranked them by their sort of ability to explain the pattern across our phylogenetic data sets. And sure enough, when we did this analysis, we find the Olson pattern of minimum temperature affecting plant height, which then affects vessel diameter. But we also found a direct effect of temperature on vessel diameter in excess of that effect through height that was also expressed this interaction with phenology and phenology by temperature. And so this kind of combined the two papers. This is the Olson paper, this was our paper, and they're both happening to determine plant growth forms. In particular, we were also interested in how this interacts with another possible trait that we kind of ignored in our previous analysis, and that's the growth form. When plants can evolve to potentially become herbaceous rather than smaller vessels or any of these things. And we know that these are nested within our traits, and we kind of only analyze woody species, right? And so we included growth form in our analysis as well. And it turns out that it lies here, where it has a weak effect perhaps from vessel diameter, but overwhelmingly it's affected by height. This is not surprising. Did you all know that trees are tall, right? So that's going to affect whether things become woody or herbaceous, right? But when you think about this, a lot of this time, this stuff kind of becomes obvious when you put it all together. But look at how different that is from the models we use when we do like an ancestral state reconstruction of woody and herbaceous, where we treat all woody things as the same woody level of woody, and that they all have an equal probability of transitioning to herbaceous and all have an equal probability of transitioning back from herbaceous. Well, if you know the height of a plant, you shouldn't think that. Tall things are not going to be able to become herbaceous overnight, and short things aren't going to be able to become woody overnight, right? And so this is pretty far from the models we use, even though we have all this other information that to any biologist kind of makes some sense. Like, it makes sense from our understanding of cause and effect. And I think this is, you know, if I think about how this applies to someone like Suman's work, so he has this, his focal study system is in the Rubiaceae. It's a group of plants that Hediotis that exists in Southeast Asia. There's a few lineages that exist in North America as well. And he was really interested in kind of these transitions to woodiness that seem to be associated with tropical mountains. And he did this analysis, and there's these kind of three clades that come out as having really high diversification rates. All of them show this transition from this normally what's a really small plant, herbaceous plant that's really drab and boring, to becoming these large trees on tropical mountains. And it's happened, three instances, it's been associated with this rapid diversification of lineages. But what's interesting about this when we actually test it at the comparative scale, whether or not you find this as significant or not kind of depends on what taxa you include. Because it turns out that not every time that these lineages become woody, you can kind of see here, do they diversify? Sometimes they don't, right? Other thing is that not all the lineages that are on tropical mountains are diversifying faster. Some of them don't, right? So depending on which taxa you include, the entire signal is just driven by these kind of three events, right? Is that enough for you to believe that those two traits are causal for driving an adaptive radiation or not? And so that's this kind of summary of that. Not every woody group diversifies faster. Not every mountain group is woody. The significant relationship depends on how many and which groups are included. And the signal is driven by a small number of groups. And I think this is pretty common in comparative analyses. Is this good evidence for cause or not? And this is an even more dramatic example that I like to use because, at least in my mind, and you can make your own judgment, I actually think that it's really strong evidence for a cause, but it's even worse because it's only one event. So I wasn't involved in this study, but this is a really nice study by Markey et al. Adaptive radiation, evolution in Nectarivory in a large songbird clade, the Melophagidae. And so these are Australian birds that have this transition to Nectarivory here on the phylogeny. And you can see as soon as that happened, there was this rapid increase in body size evolution. The length of the branches indicates the rate of body size evolution. And so that one event seemed to trigger this massive change in the body forms of these birds as they colonized a bunch of niches, presumably. But if you look at this pattern more closely, you'll see that there were several transitions to Nectarivory that were not associated with the shift. And there were several reversions back to non-Nectarivorous that even had higher rates of body size evolution than the initial one. And so what are your statistics supposed to do here, right? And yet I can't help but look at this pattern and think, dang, that Nectarivory must have had a role in causing this diversification of body sizes, divergence in body sizes at that point, right? So what's missing here? And this is kind of a more enters into a more speculative part of my talk here. And this is something I've been thinking a lot for a long time about how we study adaptive radiation, what we think the causes are of adaptive radiation when we do macroevolutionary studies. And so, we're interested in explaining exactly these sorts of things where, you suddenly get a whole bunch of different body plans and species versus diversification that may not be associated with this adaptive change. And this all kind of goes back to a dissertation project that I had, where we looked at combining micro and macroevolutionary data across scales of time. And so we collected all the data we could get on body size, linear change in body size measurements from microevolutionary data, fossil data, and tree of life data. We put them together and they all fit together really well. But what was remarkable about this pattern was this band going from intervals of 1 year to 1 million years. So this is the amount of change you expect for evolution. This is all in animals, and it's from all the studies we could get. So the simplest you could get, time for evolution and the change in body size. And what this plot says is that the amount of change you expect after 1 year, 10 years, 100 years, 1,000 years, up to 1 million years is the same. So it doesn't go anywhere. You see stasis under 1 million years, and that most of our macroevolutionary rates are bounded to be million-year rates. Change happens or accumulates on million-year timescales. And we know evolution can do so much more, like here, artificial selection of dogs' body size. I put it here, it probably should be more like 300 years when most of these changes have happened, but I put it way back at the very first domestication of the dog. And you can see that evolution can produce this point, and if it can produce this point, you can blow up really fast. It could produce much faster rates, but we rarely ever see that. And so this is what's called the paradox of stasis. We know that evolution at the microevolutionary scale is the response to selection equals the additive genetic variance times the selection gradient. And this equation tells us that if we actually go out and measure each of these quantities, what we see is that change is often rapid, R is high, genetic variation for traits like body size is ubiquitously high, and selection is often strong as persistent. We measure natural selection, beta is often very large, and yet when we multiply that by time, we get stasis. And so, this led to me to thinking about why do we see million-year time scales? We did an analysis that suggested that we also observed burst-like evolution, that it took things kind of maintained in this band for 1 million years, and then after that you could see these kind of bursts in evolution at the million-year time scale. And whatever explanation we have for that, some of you may be already thinking of some explanations that make sense to you. What I always thought was, in order to explain this pattern, it has to be something that's enables some stability to this pattern. Why do we always see million-year rates, no matter if we're studying foraminifera or we're studying elephants? There's a lot of things that I don't know much about foraminifera, admittedly. You guys, probably many of you in the audience do. But I know there's a lot of differences between them and elephants, right? And yet, if you look at time series of change or look at evolutionary rates in things like body size for these two groups, they're really not far off from each other, right? And it's hard for me to believe that the selective environment or many things about these things are in common. So any explanation that would kind of unite all these different organisms has to be stable to all the differences that they have in their biology. So the typical explanations for this stability are things like stabilizing selection, that maybe there are hyperstable niches that just exist out there in nature, or maybe speciation is something that also takes millions of years. It takes millions of years to evolve incompatibilities. You need speciation in order to start accumulating this divergence, accumulating this trait change. Some of you may be thinking of punctuated equilibrium. And one of the hypotheses that I think is closely tied to punctuated equilibrium is what's called the ephemeral divergence model. The idea that most of the change we observe over short time scales doesn't end up going anywhere. It's ephemeral. And this is a hypothesis that's championed by Doug Fatuma, and I think Stephen Jay Gould kind of accepted as kind of the mechanism of punctuated equilibrium in the years before his death. And I really agreed with Doug on this, and I thought we agreed, but then we had a conversation in his office when I visited Stony Brook, and he grilled me for an hour and was like, "You don't agree with me." And I was like, "Okay, this is interesting." And one of the pieces where we disagree is found in this line right here. So this is from Doug's paper. Long-term anagenetic change in some characters is then a consequence of a succession of speciation events. Okay. And so he's assigning speciation as a cause of trait change here. So does speciation cause adaptive radiations? Does it increase rates? Well, that seems like a kind of a dumb question because adaptive radiations are defined by the number of things, species. So clearly speciation is a cause of adaptive radiations. But this is a related question that I think is much less obvious to me. Can failure to speciate explain why some species don't adapt? I think that's a much harder question to answer and suggests to me that speciation may not be sufficient for this at all. So what is a cause? Well, we can take what we typically study, which is a correlation, right? Do we see more trait change given that there's been speciation, given kind of the background rates of trait change that we observe overall? And if you read the macroevolutionary literature, I would argue that this is a pretty solid pattern. We see this when things speciate more, we observe more trait change. But remember in the Simpson's paradox example, the key to unlocking a cause is to imagine doing an experiment when you can't, right? And our math doesn't really have an operator for that. So we can introduce what's called the do operator. So imagine artificially affecting speciation rate. Would that increase the probability of observing a trait change? This is less obvious to me. And so what we're seeking here is the link between speciation and trait change, and there are probably confounding factors to both of those at the macroevolutionary scale. One way that we could answer this question, interestingly enough, is by using what's called an instrumental variable, something that we know affects speciation and speciation alone. This thing could, if this instrumental variable, maybe at the microevolutionary scale that we know affects speciation, is also correlated with trait change, then it has to be acting on trait change through this path, through speciation. It allows us to estimate this arrow, even if we have confounding factors. And so some candidates for that, things like the propensity to evolve intrinsic prezygotic isolation, or maybe population structure, dispersal capabilities, those sorts of things that we know kind of should affect speciation, right? Well, when we do macroevolutionary tests of this, Dan Roboski has done several of these, looking at intrinsic reproductive isolation and speciation rates, population structure of speciation rates. It turns out that neither of these things are correlated with macroevolutionary speciation rates. And so, unfortunately, our test is kind of screwed up because not only can we not estimate this arrow, we actually can't estimate this correlation or this effect at all. And so this kind of, I think, puts a wrench in how we study speciation, how we study adaptive radiation at the microevolutionary scale, and how it's related to macroevolution. And so thinking back to, okay, what do we know from macroevolutionary studies? I would argue one of the most ubiquitous patterns is that predicting extinction risk is dependent on geographic range size, and that we observe that speciation and extinction rates are highly correlated with one another. And so given those two facts, you could potentially make this causal model where population structure and reproductive isolation does affect speciation and diversification, and that may influence trait change. But these things also break up species geographic range. They make them smaller. That may increase population extinction, which then may decrease diversification rate and dampen trait change. And this is kind of really at the heart of what Fatum was talking about with a lot of this ephemeral divergence model. And so what is a cause? This may be true at the correlational level, but if artificially increasing speciation by increasing reproductive isolation may not increase trait change because it may actually increase the risk of extinction at the same time. So then what are the causes, what could we consider as a candidate cause of an adaptive radiation? If you read the literature, the ubiquitous thing is ecological opportunity. How does that look when we actually do macroevolution tests? Well, there's this paper from 2010 written by a bunch of close colleagues of mine, actually, by Jeremy Yoder, where they talk about adaptive radiation as a flattening of the fitness surface that allows organisms to move across these different adaptive peaks and go see, different phenotypes, right? But this doesn't make a whole lot of sense to me, and I'll explain why by kind of a simulation model, an example, a toy model here to think about this, okay? And so the idea here is that you have a bunch of different populations that each have their own unique environmental conditions, and so they have their own adaptive peaks. So I'm just zooming in on two here, the yellow and the teal ones. And we have a lineage that has a particular phenotype. It disperses to the teal patch here, and then evolves to that optimum, okay? And so in adaptive radiation, this kind of seems like an adaptive radiation. But what happens if we start flattening these adaptive peaks, if we flatten the landscape, okay? Well, what happens is you take away adaptation, okay? So what I've done here is I've simulated a model where I have 100 populations that are arrayed with different optima ranging from a phenotype of -3 to 3. And we start out with a population with a phenotype of 0. And so these populations can send out migrants, they land on these new patches, they adapt to the new conditions, and if they get a big enough population, they show up in my simulation, and these guys are all adapted to their local adaptive peaks. On adaptive radiation, we're looking for an expansion of divergence. We want to get the maximal amount of divergence. There are things working against that though, extinction and also potentially swamping from gene flow. So let's start with strong selection. We get this pattern. As we flatten the fitness surface, we initially get a little bit of expansion of divergence, but then something starts showing up. And if we go to the limit of no selection, with a fully flat fitness landscape, you no longer get adaptive radiation because there's nothing pulling things to a new adaptive peak, right? So flattening the fitness surface at its limit is just genetic drift. That doesn't seem like an adaptive radiation to me. So what we're struggling here with is a language to talk about how we represent adaptive radiation in our models that we actually use. And so what parameter can we actually turn up in this model that causes more divergence to happen at a faster rate? Well, one thing that I think is missing from our models is absolute fitness and demographic constraints, which you have to consider as soon as you allow the possibility of extinction. PopGen, we don't think about this as much because we're usually focused on a single population. As soon as we go to species level, as soon as we go to phylogenies, we have to consider absolute fitness and the probability of extinction. And it's very likely that lineages that move to a new adaptive peak may start adapting, but they're going to start losing individuals as they're approaching that new adaptive peak and potentially go extinct before they get there. And so absolute fitness matters. These demographic constraints maybe can really constrain adaptation at the macroevolutionary scale, where extinction is a thing that happens regularly. Adaptive radiation then could rescue that by what I call adaptive uplift. We shift the entire adaptive landscape up so that if you make this transition and you adapt to this new adaptive peak, you have positive population growth even when you're maladapted. You're growing even when you're not perfectly adapted to the conditions. The population is doing well. This, I think, maps onto what we think of as ecological opportunity pretty well. Even if you're not a good at your job, you're still able to fill a niche because there's not any competition for it. And so what happens if we take this model? So what I'm going to have is the maximum population growth rate, and I'm going to start out with a situation where the peaks barely go above to a positive growth rate, even when they're perfectly adapted. There you get very little divergence, but as we start lifting up these peaks, we just keep getting more and more divergence, more and more ability to colonize different peaks very rapidly, which would map onto an adaptive radiation. Now, if we take the idea that this is all with stationary peaks, what if these peaks randomly move up and down? So we can combine a simulation model where we take demography, adaptive peaks, genetic drift, population extinction, and gene flow, and I'm going to take quantitative genetic models where I use priors from biologically informed distributions, what we believed were realistic parameters for these things. And we allow these peaks to just kind of move around randomly. We seed our population, it starts adapting, and a really interesting thing happens. We ran this for macroevolutionary time, and it fits very well with our empirical pattern of stasis on million-year timescales. And what was really interesting is that these things did not stay stationary on the same adaptive peaks, but the population moved among adaptive peaks in a way to avoid evolving. Because the populations that did the best were the ones that were already adapted. They had this high population growth rate, they sent out lots of migrants, and they colonized peaks preferentially that were close to their existing phenotypic optima, which dampened the effect of the changing optimum itself. And so we did basically an approximate Bayesian computation using our biologically informed parameter distributions and selecting only those simulations where the simulation fell within our empirical distribution, and saw what subset of parameters, what parameters were important for kind of imparting stability on this process. So these simulations created too much divergence for our empirical pattern. These ones fell right within our pattern and had this sort of stasis. The most important parameters for determining this were the demographic parameters, in particular the population growth rate, the absolute fitness. In fact, the absolute fitness had to be almost at 1 in order for you to get this pattern, that it matched the empirical distribution. Things like adaptive landscape width didn't really matter. Carrying capacity had a slight effect, and heritability or additive genetic variance had a very weak effect. Only Rmax values that were near replacement. In other words, when you are perfectly adapted, you're barely growing your population, only those produce long-term stasis. What could cause this sort of pattern? Well, what makes sense to me is going to the ideas of ecological opportunity, that it's biotic, coevolution, and the Red Queen effect. That even if you're perfectly adapted, over most evolutionary time, you're barely able to sustain a population at current levels. Any sort of change will take you down from that, right? And potentially cause extinction. When you get this situation, you get million-year stasis. Where you can escape that temporarily is if you get this peak uplift, which can then trigger a temporary adaptive radiation until biotic interactions catch up to you and potentially take you back down to the normal rates of evolution at million-year timescales. So if we think about what causes adaptive radiation, I think that this is a good candidate, because if you think about artificially increasing absolute fitness, you increase the change in the trait, you also decrease the risk of extinction, you allow adaptation to these distant peaks, and you also potentially increase the speciation rate. So these are all effects of absolute fitness of a lineage rather than causes themselves. And I think this is important for how we study these questions, because we typically take one trait at a time rather than thinking about the combination of a key innovation and new biotic interactions. So rather than looking for a burst of trait evolution or diversification with every instance of a key innovation, most of the times those are evolved in coevolutionary arms races and they don't actually come to an advantage. What might trigger an adaptive radiation is when you take a key innovation combined with a new biotic context, a dispersal to a new area and dispersal to a new habitat, where that key innovation can give you a temporary advantage. And that is, I think, what we mean when we say ecological opportunity. And we might be able to infer where ecological opportunity is by combining our hypothesized innovations with this new ecological, new biotic context, which would allow those things to be a temporary advantage to trigger an adaptive radiation. And this is a pretty harsh hypothesis because you're predicting exactly where on the phylogeny those shifts should occur. And if you're wrong, then you probably don't understand what the triggers are for adaptive radiation in your group. If it lines up perfectly, then that's a pretty strong evidence that you're getting at what's happening. And so I think these things are a little more complicated, but I think the ultimate driver of all of these things that we're studying is potentially has been overlooked in phylogenetic comparative studies, and that's this kind of absolute fitness ecological opportunity that's in our verbal models but hasn't quite made it into the actual statistics that we do. So the implications of this is that peak uplift is the predictable result of ecological release, which I think the evidence for that is from mass extinctions, introduced species, and classic adaptive radiations. Million-year rates are regulated by the red queen, which regulates absolute fitness over long timescales, and that this kind of geographic range scale sets the pace of this kind of red queen evolution and macroevolutionary rates. And we see from our simulation that habitat tracking, kind of similar to the salamander-Bogart effect with phenology, emerges as this kind of consequence of adaptation across the geographic range. And so hopefully I've convinced you that to some degree that causal thinking can do a lot more work than mountains of data sometimes, and that we can integrate more of what biologists know. Maybe we can, about cause and effect, we can do a better job of understanding these macroevolutionary patterns. And so taking a page out of kind of the growth of phylogenetics, I think that the phylogenetics was really kind of spurred on by integrating our knowledge of population genetics and molecular evolution to understand trait evolution and phylogenetic comparative methods. I would argue we need to incorporate more knowledge of cause and effect from all the fields where we have knowledge of these relationships among traits. And with that, we can keep growing phylogenetic comparative methods and learning, even when we are faced with the situation where we only have these correlative patterns in our data. So with that, I'll like to acknowledge my collaborators in my lab and can take questions if there's time.
Great, thanks very much.
Thank you.
So it looks like we have just a couple minutes left if anyone has questions. So one thing that struck me that you said, Josef, was this idea about the changing fitnesses being a component of our verbal models, but not our not our quantitative models. And like that really hits home for the early thinking about adaptive radiations here in Hawaii, where there's a lot of talk of like, you have these founding events and then relaxed selection because there's no competition, which is basically the same thing as saying the fitness is changing.
Mm-hmm.
So I'm just kind of digesting that at the moment. So maybe my question isn't very clear.
Yeah, I can say a few words about that while you're thinking about it. So I think that, at least for me, what kind of was restrictive to my thinking about this was kind of in the population framework, only thinking about relative fitness. And the only way you can weaken selection in that framework is by flattening the surface, right? And that's why that appeared in this paper by Yoder et al. But what doesn't make sense about that, and also, that's tied up in how we think of genetic constraints. You don't have enough genetic variation. Well, you have a lot of genetic variation for almost all these traits all the time, even in founder populations. There's some limits to that, but for things like body size, at least, that's fairly true. And so, I think that the limitation was kind of imposed by thinking of the pop-gen world. And if you start thinking about, where you now have populations that can go extinct and your units are populations and species, now you have relative fitness isn't good enough. You have to have the fitness across all of these things. And so I often include like a slide of like geckos that are pollinators, right? A gecko is clearly not a good pollinator. It's not on an optimum for a pollinator, right? Like hummingbird is obviously a much better optimum for being a pollinator than a gecko. But geckos on islands do this, right? And they're not particularly good at it potentially. They may not be the best at their job, but they're still able to survive because they have ecological opportunity. There's not other species that take their niche and, the flowers aren't trying to exclude them from the nectar, all this stuff, right? And so that's sort of the model, right? Is that even the valleys are lifted up and the peaks are lifted up. It's not that the peaks are flattened, right? And I think that fits pretty well with the way we think of adaptive radiations in places like Hawaii, where it's, everybody gets to survive, even if you're not particularly good at whatever niche is available, right? Until you get there, right? Yeah, Rosanna?
Joseph, thank you. It was very informative, and I'm thinking about some examples that fit also this that are not adaptive radiations. But one of the things that came to mind when you were talking about this lifting peaks was this paper by Dan Grabowski, where he argues that, for example, carrying capacity is a limitation for speciation and can actually induce extinction. And I think that paper was highly criticized because we think about, population parameters not moving at the scale of macroevolutionary changes, right? So how can you say that a carrying capacity affects the number of species in the long term? But is there a way, like it seems that from your example, carrying capacity could move those peaks down, right? Instead of up.
Yeah.
And I don't know if you have thought about integrating that. And
so it is definitely a parameter that comes out as being significantly shifted in our kind of ABC analysis, right? Like it's different from the prior. Like bigger carrying capacities will induce more of this pattern, and so it is important, and it's more important, surprisingly, to then things like genetic variance, right? In determining this, because these demographic constraints are so important at this scale, right? So if you can resist, and actually I think it's maybe opposite than that, because the bigger the carrying capacity, then the more inertia there is. You get these kind of populations that are on these in these lineages that aren't changing, and they'll kind of just sit there, and they'll send out lots of migrants, they'll swamp out, and this is kind of you can have this, if things are changing all the time, paleontologists typically think, or population geneticists, if selection is really strong and you have large population sizes, selection is going to be very efficient and change might be very fast as well, right? That's kind of the opposite of what you hear from the paleontological literature. Big range, big population sizes, that's those lineages that have super strong stasis, right? Why is that if things are changing? And I think that the difference between those is you have to think about now a heterogeneous landscape over a geographic range, over this kind of habitat shifting with these demographic constraints, and how that plays out over macroevolutionary time, which I think is kind of a missing piece when we scale from micro to macro.
Okay, so we should probably call things there. Let's just thank